My lab has been working on the HIV replication and myeloid cell, mainly non-dividing cell, just because the cell biology and biology endpoint is quite different from replication in CD4 T cell. It's almost opposite, and so I will explain a little more detail. So the understanding of HIV replication in non-dividing cells is actually recently very expanded very much due to discovery of myeloid cell specific restriction factor called SAMHT1 that Mario already explained. So very brief, fast background. HIV infected two different cell types, activate CD4 T cell and a non-dividing macrophage. And however, HIV infected T cell showed a total different outcome compared with HIV infected macrophage. So HIV infected T cell undergo G2 arrest and then quick cell death and eventually lead to loss of immune protection. Some of them survive and then become memory T cell, and then through the reactivation it contribute to HIV persistence. And however, HIV infected macrophage, unlike a T cell which quickly dying, macrophage actually live longer, like Mario said, and becoming long-living viral reservoir, and also contribute to HIV persistence. And also tissue macrophage, such as brain macrophage, microglia, also cause very complicated problem in HIV patients called neuroAIDS. So cell biology outcome is opposite. One die quickly, one live long, and also virology outcome is also different. HIV replication and activate CD4 T cell is very fast. Within few days, there's a lot of virus coming up, but HIV replication and HIV-1 replication in macrophage is very delayed, kinetically suppressed. So this almost opposite, like I said, and between these two cell types in terms of HIV-1 replication. So about 10, 15 years ago, we were very interested in understanding how HIV replicate in macrophage purely because macrophage is non-dividing cell. There's no support for DNA synthesis, terminally differentiated non-dividing cell. But HIV has to make a DNA during reverse transcription. So the question is how HIV overcome the lack of support from the cell they are infecting, particularly in macrophage. So as you know very well, and then HIV retrovirus convert the single-strand RNA to double-strand DNA. This is a DNA synthesis, DNA polymerase step, requires several components, and reverse transcriptase, there's a viral DNA polymerase we call RT, and then template, viral RNA genome or viral DNA, and then you need a primer to initiate the DNA synthesis, and most importantly, reverse transcriptase require substrate, the building block of DNA, that is DNTP. And as enzymologist, it's clearly rate of reverse transcription of HIV-1 is totally depends on the availability of DNTP in the cell they are infecting. And the reason I said ask this question is because the DNTP concentration varies significantly depending on the cell type. Many dividing cell versus non-dividing cell, because DNTP biosynthesis is a closely tied cell cycle. So activate CD4 T cells, dividing cell, fast replicating. There's chromosome replication, so there's abundant DNTP in the activated T cell. But terminally differentiated macrophage, we assume that there is almost none because the cell don't need all DNTP for DNA synthesis. So about 12 years ago from today, we asked a question whether there is sufficient DNTP in a non-dividing terminally differentiated primary cell. So my student, Tracy Diamond, developed an assay that allowed to determine the concentration of DNTP in activate CD4 T cell and macrophage, and also confirm through the collaboration with the Shinazi group. And indeed, activate CD4 T cell has about 100-fold times higher DNTP concentration than macrophage. So it's not really surprising because we already kind of predicted with non-dividing cells should have a low DNTP concentration. But actually surprised the actual concentration of DNTP in macrophage, that's 20 to 40 nanomolar. This is almost none in terms of DNA synthesis, as you know very well, and the most, PCL has a 250 micromolar. So this is almost 100,000 times lower DNT concentration that we usually know. So then the question is how HIV replicates synthesized DNA in macrophage where there's no DNA synthesis and there's not much DNTP around. But throughout the study, we already know the host cellular DNA polymerase, their Km value is right in the middle of DNP concentration found in dividing cell. So you can tell there is a evolution between enzyme activity versus substrate availability. So we ask whether HIV reverse synthesis also works in the low DNP concentration, unlike other DNA polymerase, because they has to work in the non-dividing cell where there's not much DNTP. So indeed, we found HIV-1 RT-Km is very close to DNTP concentration found in the macrophage. So, throughout many studies, we concluded that probably evolution happened that HIV RT worked very efficiently, even low DNTP concentration, in order to support the DNA synthesis in the non-dividing cell, such as macrophage, where there's not much DNTP around. So the conclusion is limited DNTP fold is non-dividing specific, myeloid specific restriction factor against HIV-1. So while we are studying this DNTP and RT kinetics and like Mario's group and many group actually working on the HIV replication in HIV-1, the replication of HIV-1, HIV-2, and SIV, and non-dividing cell and macrophage. Like I said, HIV-1 replicates very poorly, very slowly. Kinetic is very suppressed. But HIV-2, you see the robust replication, almost like a T cell. And that's due to, Ron also talked about that, and then because of VPX, HIV-2, many SIV strain encode viral protein X, VPX, that allow them replicate very fast, even in the macrophage. So about 5 years ago, there was a back-to-back paper done in Yasek in Cleveland and Montserrat in Montpellier in France, and published the SAMHT1 is the factor, host factor, myeloid-specific factor that suppress viral replication. And then this SAMHT1 restriction factor counteracted by VPX. So in HIV-2, SIV infected macrophage, they release VPX, bind SAMHT1, degraded SAMHT1, and then let the HIV-2 SIV to replicate very fast in non-dividing cell. So but we didn't know the mechanism yet until a few months later. John Stoy's group and Michelle West's group in London published SAMHT1 is an enzyme. It's an enzyme that's not enzyme. It's an enzyme that hydrolyzes DNTP. So basically, cleave off a triphosphate from DNTP, generate the nucleoside and triphosphate. And so after this study, we proposed that probably SAMHT1 is responsible for such a low DNTP concentration that we found in macrophage, because SAMHT1 is quite abundant in myeloid cell. So we test that. So idea is that because SAMHT1, DNTP is all hydrolyzed and in macrophage, so very low DNTP concentration. But when the HIV-2, SIV infected, it releases VPX, counteract the SAMHT1, and possibly elevate the DNTP concentration above the Km level of RT, so then replication of HIV-2 and SIV can happen rapidly. That's what we found. So when you treat the macrophage with the virus-like particle containing VPX and without VPX, you actually see the elevation of DNTP above the Km level of RT, and that support the idea that yes, VPX is elevate DNTP concentration and then accelerate the proviral DNA synthesis in non-dividing cell. So this paper is our summary paper. So basically, in macrophage, when you treat the macrophage with VLP containing VPX, you see the rapid degradation of SAMHT1 in right side, and then followed by rapid elevation of DNTP, and then accelerate the proviral DNA synthesis and reverse transcription of HIV-1 in non-dividing cell. And that's probably what happened in HIV-2 and SIV when they carried VPX in the non-dividing cell. So I'm going to summarize a few studies we did, and throughout the very extensive collaboration in many different groups in terms of HIV and SAMHD1. So turns out SAMHD1 also restricts HIV-1 in resting T cell, which is very important for transmission. And also, it turns out that SAMHD1 is very important antiviral activity in the host. When you knock out the SAMHD1 in mouse model, it turns out it loses antiviral activity, and by elevating the DNTP concentration, and then there's a certain components and that push the host to protect them from viral infection. So there's some genetic data. SAMHD1 is important in viral control in mouse model. And turns out that HIV-1 is not the only virus infecting myeloid cell. There are a lot of double-stranded DNA virus, such as herpes virus, pox virus, also infect the myeloid cell, non-dividing cell, and turns out SAMHD1 also control this double-stranded DNA virus. But interesting part is this double-stranded DNA virus, like herpes virus and pox virus, they have their own DNTP biosynthesis machinery, like ribonucleotide reductase, thymidine kinase, but still SAMHD1 can control this double-stranded DNA virus in non-dividing cells. So you can see that there's an arms race between host and myeloid cell infecting pathogen for competing their building block of their genome, which is DNTP throughout the evolution. And a few months ago, we also reported through the collaboration that SAMHD1 also control hepatitis B virus, obviously because Hep B require DNTP during the reverse transcription in liver cell. And so there are lots of other studies that expand the role of SAMHD1 for controlling viral replication. But another twist of story is that SAMHD1 also has another activity called RNase and DNase activity. It turns out it's very also important antiviral activity of SAMHD1 by targeting or escaping nucleic acid sensing innate immunity system. So there's some other mechanism that we probably don't know yet about the SAMHD1 in terms of antiviral activity. That will be very exciting in the next few years about this new activity of SAMHD1. So, summary, when HIV-1 don't have VPX infect the myeloid cell, SAMHD1 suppress the reverse transcription by depleting the NTP and restrict the viral infection. But HIV-2, SIV bring the VPX, binds SAMHD1 and the proteasomally degrade SAMHD1 and elevate the NTP concentration, and then allow the HIV-2, SIV to replicate rapidly in even non-dividing cell. And while we are doing this collaboration, extensive collaboration, we had also alternative pathway, alternative hypothesis about HIV-1, because HIV-1 doesn't have VPX. Then how does HIV counteract SAMHD1? And Mario already talked about a little bit, but we had a little different alternative pathway that probably HIV-1 evolved to counteract the SAMHD1 away from VPX. That is, maybe HIV-RT reverse transcriptase become very efficient and evolved to be very, enzymatically work very well, even low DNTP concentration. And possibly reverse transcriptase of SIV, HIV-2, which have VPX, they probably didn't have to evolve to be efficiently work, even at the low DNTP concentration. So that's our prediction that RT of HIV-1 RT doesn't have VPX probably works very well, even low DNTP concentration in myeloid cells, unlike RT of SIV. They don't have to because VPX elevated the DNTP for them. So we purified many RTs from HIV, 5 different clade, and SIV, many different clone, and then measure KM and Vmax value. And as you can see here, all the HIV-1 RT, their KM value is very close to low DNTP concentration found macrophage. But many of SIV RT show much higher KM value, close to above the or near the T cell DNTP concentration. And however, the Vmax, KCAD, still remains same. So overall enzyme and efficiency in the HIV-1 RT has much better than HIV-2 RT. And so this idea to support that probably HIV-1 RT use RT, not VPX, to somewhat counteract the SAMHT1 and in order to support the DNA synthesis in myeloid cell. So this is final summary. So then HIV-2 and HIV infect the myeloid cell, and SAMHT1 comes, and then suppress reverse transcription, VPX come, and then degrade SAMHT1, elevate the NTP, and then replication can happen very rapidly. But HIV-1 doesn't have VPX, and so when the HIV-1 infect the macrophage, SAMHT1 comes, suppress reverse transcription, but possibly RT evolved to be, we call super RT. Enzymatically a little better than normal RT that we know of. And then that allowed, or that weakens the selective pressure coming from SAMHT1 that allowed the HIV finish proviral DNA synthesis, even very low DNTP concentration in non-dividing cell. So I list, so that's pretty much all my talk, and this is the kind of question that I listed, what we want to know and what we have to answer in terms of HIV macrophage. Maybe we can talk about that in the later and today for discussion part. The main question that we are interested in the lab is why lentivirus evolved to infect a non-dividing cell. Non-dividing cell is a horrible cell to do because it's metabolically very inactive, and there's no support to many of metabolic ingredient, transcription, replication, anything. But why HIV or lentivirus evolved to infect non-dividing cell? Why develop the fancy VPX, VPL like a protein to achieve the non-dividing cell? And then Mario said exactly the same thing. Is HIV macrophage infection is contribute to pathogenesis? I really don't know. Hopefully we can answer that. And also blocking HIV myeloid infection is essential for HIV cure. Well, David Ho said it's about 1 to 5% in circulating viruses from non-dividing cell. It's important to remove final 1 to 5% of virus from the non-dividing cell for cure. We don't know. And also, as you know very well, all the myeloid cell is in the tissue and organ that known to, in the place that's very difficult to deliver any antiviral. So how we can deliver antiviral to myeloid cell? And the last question we want to answer is HIV-2 is not really pathogenic compared to HIV-1. But HIV-2 replicates very well in macrophage. So that doesn't make any sense in terms of a conventional virology theme that if virus replicate fast, usually more pathogenic. But that's not the case here. So we're missing something, and maybe HIV-2 replication in macrophage fast, and it's not really a good thing for a virus. So we have a couple of hypotheses for that. But anyway, this hopefully some of them we can discuss this afternoon. So this is all the lab member, previous and current member contribute to our work, RT work and SAMHT1 work, and many collaborators, and especially Ray Schneider's group, very intense collaboration for study SAMHT1 and macrophage infection. And this is NIH support. And with that, thank you very much, and then I'm happy to answer questions if you have time.